Welcome back to Friday Focus. So in this video, we'll be discussing the exercise questions given at the back of the chapter, the fundamental unit of life. So here I have used the word discussion because I truly trust students that they can think and reason on their own and they can arrive at the answers on their own once the right kind of guidance is given, right? So I will in this video, I'll take you to a point where you'll be able to find the answers yourself. And still, if you need help, I'll also mention the reference. Okay, so what are we waiting for? Let's begin. Before we get into, start answering the questions and start discussing the answers of the questions, I'd like to mention the format. I'd like to explain the format that I'll be following. Firstly, I'll be reading out the question. Next, I'll be suggesting or the hints, or I'll give you the leads, maybe the key words that'll be like aha, that'll ring the bell in your mind, and then you'll be able to answer the question yourself, right? So those hints and keywords will be discussed, and finally I will also mention which one of my videos you can look up at what time interval, so that you can learn more about it, get the concept, and simultaneously a link will also appear that you can click and go to that particular video and clear the confusions right away, right? So if nothing, you can also look up the references, and I'll be mentioning them in the description box below. And yes, at the end of the day, you can always go back to your textbook as well to find out all these answers, right? They'll be hidden there somewhere. Anyway, so firstly, let's start with the first question. First is, make a comparison and write down ways in which plant cells are different from animal cells. So here basically you have to differentiate between plant cells and animal cells. So bring to your mind the pictures, the diagrams. So if it is asked for as a 9-mark question, you'll obviously need to draw the diagrams as well. So we have been looking at the diagrams since, in all the videos that I have uploaded so far on fundamental unit of life. So bring those to your mind and then compare. This is basically the summary of the whole chapter, right? So what are the features in which they differ? Firstly, they differ in their size generally, okay? And then cell wall is a major point of difference, right? In one, there are no cell walls present. In the other, there must be cell walls at any cost, right? And then plastids, again, it's present in one kind, it's not present in another kind. And also the vacuoles, the way in which they appear, the number, the shape, the size, all differ in plants and animals. So I hope now you have remembered the points of difference, and now you can answer on your own the question, elaborate more on these points. If you still need help, you can always look at the video, the part 7 video of this series that I have made, and check it out at the time interval 4:40, link above. And in that I have given in a tabular form, I have compared plant and animal cells. Now you won't find that in your textbook, right? Now, secondly, what is the other question? The next question, how is a prokaryotic cell different from a eukaryotic cell? Now, the term itself is a clue. Pro means in biology always primitive, and karyotic is derived from the word karyon, which means nucleus or center nucleus, okay? So, primitive nuclear region, how was it? And modern nucleus, advanced nucleus, how is it? So that is the main difference between the prokaryotic and eukaryotic cells. So how else do they differ? They differ in their general size, nuclear region as we already discussed, and the main ingredient, the main part of the nucleus is the chromosome. So they differ in the chromosome number as well, and also in the presence and absence of membrane-bound cell organelles. Those are present in one, not present in another. So, based on these characteristics, try to answer the question. Okay. if you still need help with that, you can check out the part 3 of Fundamental Unit of Life series done by me. And in that, the time interval 1952. Then the third question, what would happen if the plasma membrane ruptures or breaks down? So in all questions, what would of this kind, what would happen if cell organelle X, cell organelle Y, okay? That is indirectly asking about the function. It needs to know what role this organelle plays inside the cell. Right? If we need to know the significance of something, we need to know what role it plays, right? And only then we'll be able to answer. If it's not there, what will happen, right? So here the subject of question is plasma membrane. So what will happen if it is destroyed or it is not there? So just remember, what is the function of plasma membrane? The main characteristic is the selective permeability. It helps in the transport of substances in and out of the cell. And how does it do so? By its selective permeability. That means it is actually the guard, which allows substances that are necessary and does not allow harmful substances into the cell, right? So this will be lost. So you have to imagine what will happen to the cell and then write the answer. So if you need help with that, part 2, 736, right? So in that I have also shown the ultrastructure of plasma membrane. That means how does it actually look if you really magnify it and see. Right? And I've also clearly explained what selective permeability is, right? So you'll be able to answer the question much more better if you see the video. Then what would happen to the life of a cell if there was, again, it's the same thing. So what role does a Golgi apparatus do? I mean, it's asking what would happen to the life of a cell, if there was no Golgi apparatus. So we already saw that Golgi apparatus is part of the cellular membrane system, right? So that means it's the part of, it functions sort of like a road for the transport of substances inside the cell to different places and also from inside of the cell to the outside of the cell, right? So the road is lost. And second thing, if Golgi apparatus goes, there is no road. And then secondly, what else is the function of other functions of the Golgi apparatus? They are the packers and movers, I said, right? Storage, packaging, modification, transportation of proteins and lipids and all this, right? So organic substances. So all this will be interrupted. Now the main function of the cell itself depends upon all these organic substances. Now depends upon the synthesis and movement, transport of these substances. So if that itself is interrupted, what will happen to the life of a cell? You can imagine. So if you need more help with that, part 5, 707, right? So anyway, but then if you want to learn more about Golgi apparatus, look at their beautiful pictures under the electron microscope to see that video, okay? Then which organelle is known as the powerhouse of the cell? Now, powerhouse, okay, what does a powerhouse usually do? It generates power, it generates energy. Now, which organelle is the organelle responsible for generating energy for the cell? Mitochondrion, energy currency, ATP. So, that is why, because it generates electricity, it's called the powerhouse of, because it generates energy, it's called the powerhouse of the cell, right? So, in case this is this question is asked for a lot of marks, you will need to explain it much more specifically, about the mitochondrion. And you might need to bring along ATP, energy currency, all those terms inside as well. So if you need to know more about the functions of mitochondria, how they look and all that, you need to look into the part 6 of the series at the time interval 0350. I'll just mention, I forgot to mention one thing. If you want to, I'll just show you the screenshot of this video. This is the video in which I am explaining the clear comparison between a plant cell and an animal cell. So in this video, if you need to get into the comparison of plant and animal cell, just in its description box below, you can see I have given the time intervals. at which each topic is discussed. So here at 4 minutes 40 seconds, you can find the comparison of plant and animal cells. So each time you feel like, referring any of my videos, just click on the link and then go to the description box, check out which topic you want to learn and fast forward it to that time and you can see it, right? Yes, now coming back to our questionnaire, number 6. Where do the lipids and proteins constituting the cell membrane get synthesized? So the cell membrane, we have learned it is a bilipid membrane, so it's got 2 layers of lipids and it's got some proteins, channel proteins and all attached on surface. So that is the structure of the membrane. And mind you, not just the structure of the plasma membrane, but also of the structure of the individual membranes of every cell organelle. All cell organelles are membrane bound, remember? So all their membranes have got this structure, that is the lipids and proteins in them. So where does all this get synthesized? Okay, the main unit that helps in the synthesis of proteins are the ribosomes. So can you remember which organelle has got a lot of ribosomes lined on its surface? It's practically called rough because of the presence of ribosomes. Okay, rings a bell? Yeah. So that organelle is responsible for creating the proteins for the membrane biosynthesis. Remember where we learned this? Under which organelle? Right. So that organelle is responsible for creating the proteins that constitute the cell membrane. Okay. And what about the lipids? Okay, its cousin. Synthesizes the lipid. So if you want to learn more about membrane biosynthesis and which organelles help in it and about the organelle itself, check out the part 4 video of my fundamental unit of life series, and at the time interval 14:30 you will find the answer. Okay, now question number 7. How does an amoeba obtain its food? The direct answer is endocytosis, but you will have to explain what endocytosis is, right? So in that explanation, you need to say which organelle or which part of the cell helps in endocytosis and what exactly it is. So what is it? It's the process in which the amoeba engulfs, engulfs. That means if this is the amoeba, my hands are the amoeba, it'll hug, and this is the food, it'll hug the food like that. It engulfs it and thus ingests it. That's how it takes in food and eats it, right? So that is called as endocytosis. When an amoeba engulfs the food with the help of a cell organelle. What is that cell organelle? With the help of a part of the cell. What is that part? You have to mention that part. And then which part do you think will help amoeba in engulfing the food? Okay, so you have to mention what endocytosis is and which part of the cell helps in endocytosis, right? So if you need more help with that, check out my part 2 video, Fundamental Unit of Life, at the interval 25:38. And before you go there, also know that in the same part at 26:24, you can actually see the video, a microscopic video of an amoeba eating 2 paramecia, and it is a very interesting video, so you can check that out. Okay, and then question number 8, direct question. What is osmosis? Whenever osmosis is to be described, we have to include 3 points. That is, we have to say that it involves the movement of water and it involves the presence of a membrane. Actually, the presence of a selectively permeable membrane, and it also involves a concentration difference of the water. The water concentration must be different. There must be a water concentration difference between where the water is going to move. So these 3 points must be included in the definition of osmosis. And if you just spend 5 seconds in this, you can get the answer yourself. Okay, so if you need help, you can go to part 2, 12:56. At 12:56 time interval, the osmosis definition is given like crisp and clear in that video. Okay, now coming to question number 9. Okay, carry out the following osmosis experiment. So again, we have to think in terms of osmosis. Okay, so take 4 peeled potato halves. Okay, peel potatoes, half them and scoop each one. Scoop with a spoon each one out to make potato cups. So if you scoop out the center, the core of the potato, you'll get a cup-shaped potato. Okay, so we need potato cups like that. Halved peeled potatoes, scooped out and made into potato cups. So 4 of them. Now one of these should be made from a boiled potato. Okay, put each potato cup in a, now all these cups we have put in a trough containing water. That means maybe a big tray or a long beaker containing water. So in that, these potato cups are kept. Now let's see what we do with each, what we have to do with each cup. So keep cup A empty. Okay. B, put a teaspoon sugar. In cup C, put a teaspoon salt. And in cup D, put again, that is D should be the boiled potato cup. And in that, we are supposed to put 1 teaspoon sugar. Now, before we get into the questions, let's just make ourselves clear what is exactly happening in each of these cups. Okay, in terms of osmosis. So first, it's kept empty. That means nothing is being done to it. So such a practice where a subject is kept untouched is very common in science. And that subject is always called as control, because it is necessary to have a control in an experiment, because it tells us, it gives us the tool to measure the difference in other subjects. That means it's almost like if you are starting a fitness regime, to take a picture today and keep it untouched, undisturbed, you save it. So after 3 months, 6 months, 12 months, you can compare the results, right? So that is the control of your fitness regime experiment, right? So that is the function of the control. It is there to help us measure or gauge the difference in other subjects. Okay. So that is the control. Keep cup A empty. Now in B, we have put 1 teaspoon sugar. So sugar, a solute is coming into play now, right? So in that small potato cup, if you put some sugar, okay, so we are creating a hypertonic environment to those cells lying next to the sugar. Okay, so that means a hypertonic environment is created inside the potato cup. Again, in this cup C also, 1 teaspoon salt is put. So again, it's the same case, hypertonic, whether sugar or salt, it is the solute concentration that we are increasing, right? It is a solute, whether it is sugar or salt. So B and C, hypertonic environment is being created inside the potato cup. Now in D, again, 1 teaspoon sugar is only put. But what is the difference here? The potato is boiled. So whenever living cells are exposed to boiling water, that the extreme temperature, what happens to these living cells? They become dead cells, right? And dead cells lose the capacity for osmosis because the selective permeability of the membrane is lost. That's why no plasmolysis also occurs, in dead cells of real leaf. Remember, plasmolysis occurs only in living cells. Same with osmosis. Both require the presence of selectively permeable membrane, and both won't happen unless that they are living. Okay, so dead cells, osmosis will not happen. That is forgiven. So let's now look into the question. Now that you know what are the realities that are happening inside each of your subjects, you'll be able to reason better. So now bring on the questions, right? So first one, explain why water gathers in the hollowed portion of B and C. Now remember, these potatoes are kept inside a trough containing water. That means outside there's a lot of water. Outside each potato cup there's a lot of water, right? So inside you are putting a lot of sugar. That means inside the potato cup there the solute concentration is high. That means hypertonic. Now, okay, I know this osmosis can be very confusing. I was a student myself, and I don't know, there was something very confusing while learning about this osmosis. So I'll try my best to clear the confusions, okay? In the video wherein I have thought about osmosis, in that I had made clear how we must answer the question to, to where the water will move. So that depends upon the concentration of water, right? Osmosis, to where water will move, it depends on the concentration of water. It will move from high to low. But then this hypertonic is exact opposite. It will confuse us even further. So let's say, why do we say hypertonic as, high concentration? Because we are measuring the concentration of solutes in terms of hypertonic. The term hypertonic, okay? Hyper means high. High of what? Of the tonic, of the solute. Okay, so in hypertonic solution, what is it? Any solution in which the solute concentration is high, right? So that is about the solute. The term hypertonic is given depending upon the solute concentration. But when the question is about the movement of water, take into consideration the amount of water in that. In hypertonic solution, because solute concentration is high, water concentration will be low. The solute concentration and water concentration is always indirectly proportional. That means when one is high, the other will naturally be low. So now let's consider the case here. Inside the potato cup B, the teaspoon of sugar is creating a hypertonic situation inside that little potato cup. Okay, so that means, what about the water? Inside the potato cup, the water concentration is low. So water will move from high, that is outside the potato, inside to inside of the potato cup. That's why water gathers inside the potato cups in both B and C. Second question is, why potato A is necessary? We have already answered that. Okay, control. Then the third sub question is, why water does not gather in the hollowed out portion of A and D? D, we already know, they are dead cells and they do not, they cannot undergo osmosis, right? So because that selective permeability of the membrane is lost. So that's why no water gathers inside D. What about A? Cup A is living. Still, why is there no water gathered in A? Because there is no concentration difference of water that has been created. There is no solute. Hence, water concentration also need not change. Water level need not change. Right? That was about question number 9. And still, if you want to clear even more confusions, you want to learn more about osmosis in a more clear way, you can always check out my part 2 video, and at 16:59, I'll be giving you, I have given you in that video a diagrammatic explanation which is not there in your textbook. I have given that kind of an explanation so that you can understand it very, very clearly and remember it. Okay. So those, I think personally, such diagrams help me to remember and understand clearer. So you can just check out that video and make out if you can learn, if you are able to learn it better. I hope you do. Now question number 10. Which type of cell division is required for growth and repair? Let's just hold on our by-hearted brain. Just think, okay? We know that growth and repair. Okay, first growth. If suppose my skin is supposed to grow according, I'm growing in my height or weight and I need my skin to grow, then these skin cells need to produce exactly the same kind of skin cells, right? Exactly identical cells my body would need. So then what does it mean? The chromosome number should be identical. Of the new cells also should be identical in terms of growth. Even repair. If I cut a finger or if I cut my hand, same cells I want to regrow. So that means the chromosome number should be same, right? So whichever cell division involves the daughter cells to have the same number of chromosome, that kind of cell division is required for growth and repair of the body. Now think, Which would it be? Okay, and the last part is which type is involved in the formation of gametes. Now, formation of gametes must involve the halving of the number of chromosomes. That means the chromosome number should be reduced to half in the formation of gametes. So whichever type of cell division involves the halving of chromosome number, that type of cell division is required for the formation of gametes. Now, so if you want to know more about these both type of cell divisions, and also if you want a comparison between, a tabular comparison between mitosis and meiosis to help understand better, and sometimes I think it is a frequently asked question to compare mitosis and meiosis. So all that has been discussed in my video, that is part 7, the previous video to this series, and at the time interval 10:52, you'll begin learning about the cell division. So in that video, I have also provided a very simple diagram that you can easily draw, easily understand, and the explanation is also like very simple as well as very clear. And thanks a lot. I hope you're not mad at me for not giving the answers right away, because it would have made your life much more easier. But trust me, it would have reduced your self-esteem like crazy, because the sweetness of when we arrive at an answer on our own, that triumph that you feel, that is very rare these days, right? These days, every information is available on its own. Even if, somebody over in a TV show also asks us any riddle or anything that you need to answer to get a car and all that, you just type it in and you have the answer ready made. There's no maza in that, so consider this opportunity to think on your own. Think a bit on your own. Just a bit, okay, on your own, as an opportunity to grow and, to prove to yourself that, hey, I can do this, man. Right? So I just wanted to give to you that moment of triumph where you're like, yeah, I know this. Okay. So yeah, I hope you did, receive some help. At the same time, I really hope you got your answers. Right? Anyway, so thanks a lot. Keep coming to my to this channel, and I'll be uploading a video on an education, where I'll be uploading an educational video every Friday. All right. So thank you. Have a good day.